Anyone who's spent any time studying biodiversity knows that as you go from a small sample to a larger and larger and larger area, you find more and more species. Biodiversity is intrinsically scale dependent. My name is Bill Kunin, and I'm going to be talking to you today about how you scale up biodiversity information from fine scales to regional, continental, and global scales. There are several reasons why we find more and more species in larger and larger areas. First of all, trivially, you can't have more species than you have individuals, and a larger and larger area will have more and more individuals. But also, as we go to a larger and larger area, we will also have more different habitats and more different environments, hotter places, colder places. And that again means more species. But also, as we have larger areas, it makes it easier for species to coexist. So competitors or predators and prey are more likely to coexist if they have a larger area to coexist in. It's easier to sleep together in a larger bed. Since the 1920s, the tool we have to describe the relationship between species richness and area is the species-area relationship. Even from the inception, we've had disagreements about what the shape of that relationship is. Arrhenius, when he first proposed it, suggested it was a power law. Gleason, the next year, came up with a paper saying, no, it should be a logarithmic relationship. Though there have been debates backwards and forwards. In the 1970s, we'd have thought that not just was it a power law, but it was a particular power law, the canonical power law with a slope of 0.25. That was the shape of the species-area relationship that most people believed in. Now we have bigger data sets. There's a consensus emerging that the species-area relationship, if we look across a wide enough range of scales, is curved in a complicated way. It's triphasic. It has a steep bit, and then a shallow bit in the middle, and then a steep bit in the end. Different parts of it look like different models. So the Gleason model looks a lot like what the triphasic species-area relationship looks like at relatively fine spatial scales. But the Arrhenius model looks more like what the species-area relationship looks like at intermediate scales. And then at very coarse scales, it gets steeper yet again. We can see different bits of biodiversity information enshrined in that curve. So alpha diversity, the number of species you find in a small sample of some sort, is the height of the curve at the scale of that sample. But the slope of the curve is determined by beta diversity, the turnover of species across space. Also, common species tend to push up the curve at relatively fine scales, while rare species tend to push up the curve at coarser scales. Ultimately, the species-area relationship ought to be the ultimate tool for describing how species diversity is affected by space across the full range of scales. The problem is, though, to estimate the species-area relationship takes a lot of work. We have to then look comprehensively at all the species that live in a large area, and that's hard to do. It would be nice if we could find ways to take samples at very fine scales and then use information from them to estimate the species-area relationship across the full range of scales. And that's the topic of this talk. In my mind, it is the single most important unsolved problem in spatial ecology. One of the things that makes upscaling biodiversity challenging is that biodiversity is an unusual variable. For most things, we could take a representative sample and measure the quantity of interest in each of those samples, and we could then average them out and multiply it by the total area, and that would be a good way to upscale the variable. So for instance, if we wanted to know how much carbon was being stored in an area, we could take a bunch of different small samples and see how much carbon was being stored in each square meter, and then multiply that by the number of square meters, and that would give us the total amount of carbon being stored in the whole area. Unfortunately, biodiversity is not additive the way carbon is additive. Biodiversity is subadditive. So if you had 6 species in one area and 4 species in another area, you might not have 10 species altogether. You might only have 7 or 6 or 8, because there may be some overlap in the species you had in the different samples. Biodiversity is subadditive, and that makes it hard to scale up. There are various reasons we might want to upscale biodiversity. We might want to estimate the number of species in a little known taxon or in a little known area. So for instance, we might want to understand how many species of trees there were in the Amazon. As it happens, someone recently tried to do that. They looked at different samples from across the Amazon and used methods, in this case, the Chow-2 method, to make an estimate of how many species there were in the Amazon. A later paper tried to look at how many species there were on Earth by looking at forest inventory samples from across the planet, and again, using the Chow-2 index to estimate how many species there were on the whole planet. Another reason is that you might be taking some sort of fine-scale experiment to try to understand how a different form of farmland management or nature reserve management would affect biodiversity, and then you want to extrapolate that finding to a much larger area, to a region or to the whole planet. So again, you need to upscale your findings to a larger area. You might also want to use biodiversity upscaling to try to monitor how biodiversity was changing across a wide range of scales. It's easy to see how biodiversity is changing at the scale of a quadrat or a trapping device. But if you want to understand how biodiversity in a country or a region or the planet as a whole is changing without having to resurvey the planet as a whole at decadal intervals, we could find use of upscaling methods. Or you also might want to use biodiversity upscaling to create models of how the planet behaves as a whole under different sorts of management scenarios, which is the whole point of BioDT. Digital twins require some sort of upscaling model to understand how local changes translate to larger scale changes. Those top two challenges are really just about trying to understand how many species there are in this larger area. And so all we really need to do is to estimate the top point of the species area relationship, while those bottom two challenges are trying to understand the whole shape of the curve. And so it's a bit tougher. We have reason to think that some of the pressures that we as a species are putting on the planet may be affecting biodiversity differently at different spatial scales. So for instance, habitat fragmentation. There's pretty strong evidence both from field research and from modeling that says that as you fragment a habitat, it tends to result in a decline in alpha diversity, in the species richness of each fragment. But at the same time, an increase in the beta diversity, the turnover across space. That can end up meaning either no change at all or even a net increase in biodiversity at moderate scales. So the same pressure can be pushing down the species area relationship at fine scales and pushing up the species area relationship at coarser scales. Invasive alien species also may affect biodiversity differently at different scales. There's pretty strong research looking at invasive alien plants, at least, showing how there are very strong negative effects on biodiversity. But even as you go from meters to tens of meters to hundreds of meters, the strength of that effect declines until there's having very, very little effect at all as you're talking about hundreds of meters. If we start looking at coarser scale still, there's pretty strong evidence that invasive alien species, for the moment at least, are resulting in a net increase of species richness. Certainly when we look at islands, the number of plant species or fish species seems to be increasing in most islands, and similarly in different areas of mainland, while for birds it's less strong. Meanwhile, at a global scale, there's clear evidence that invasive alien species have resulted in extinctions, which does mean that global biodiversity is decreasing because of invasive alien species. So we have the same driver pushing down species richness at very fine scales, pushing it up at intermediate scales, and pushing it down again at global scales. Climate change may also be behaving differently at different scales. So again, there's pretty strong evidence that at a global scale, there are lots of species in trouble because of climate change. But if you look at much finer scales, the effects seem to differ in different parts of the planet. So in arid zones, it does look like climate change is resulting in a decline in local species richness. But if you look at subpolar and polar zones, biodiversity seems to be increasing because of climate change. And in temperate zones, it seems to be somewhere in between, with some places increasing, other places decreasing. Meanwhile, at intermediate spatial scales, there's some evidence that species that are expanding their geographical ranges with climate change are doing so in a cohesive manner, while those species that are retreating from climate change tend to do so in a patchy manner, leaving behind little puddles of occupancy in local microclimates where they can still persist. The net consequence of this is there seems to be a pattern of expanding ranges expanding faster than retreating ranges are retreating. So at least for a transient, there seems to be a slight increase in biodiversity because of climate change at intermediate scales. So again, the same driver is causing decreases at a global scale, possibly increases at an intermediate scale, but mixed effects at finer scales. Taking them all together, there seems to be a suggestion, at least, that biodiversity change is different at different scales, with fine scale decreases in most cases, quite frequently moderate scale increases, but then global scale decreases. So the shape of the species-area relationship itself is being changed by human interventions. That makes understanding not just the shape of the species-area relationship, but the changes, the dynamics of the shape of the species-area relationship, a matter of urgency. The first paper that suggested that it was possible to upscale from local point samples to the whole species-area relationship was one by John Hart in 1999. He pointed out that we can easily take local point samples and estimate the height of the species-area relationship at one scale, the scale of the sample. But then since the slope of the species-area relationship is determined by beta diversity, the turnover between samples, we can look at the turnover between different local samples. And looking at the turnover between different samples that are different distances apart will give us that slope at different scales. And we can then knit together those different bits of slope and produce the species-area relationship as a whole. Now, as it happens, the original Hart model had some mathematical flaws in it, but in the time since then, there's been a proliferation of different methods suggested to do this job. There are many different approaches that people have taken to doing this upscaling. Some of them, like Hart's original model, look at beta diversity and how that declines with distance. Others have looked at the species abundance distribution and tried to understand how rare species can be predicted from common species. Still other ones try to fit a model of the natural world as a whole and then use local biodiversity data to try to parameterize that model. Still other ones use sampling theory and try to understand the statistics of finding species in a larger and larger sample. Still other ones just fit a curve and extrapolate that curve out to coarser scales. In my mind, these different methods come into two big categories. Some of them are idealized methods where an understanding of how the natural world ought to behave is used. The nice thing about this is you can then often parameterize that model using relatively little information and get insights across a wide range of scales. One example of that is John Hart's maximum entropy model. Other methods are descriptive. They often need much more data to fit their models, but they're also much more flexible. And as a consequence, they may be better at trying to understand how we as a species are messing with the natural world and changing biodiversity patterns across scales. One example of that is Chiara Polce's method. One method I wouldn't recommend is to fit species distribution models or species occupancy models to each species one at a time, and then simply to sum those. The sum of these occupancy area relationships produces the species area relationship. So in principle, it ought to work. The problem is that you usually can't fit species distribution models to rare species, and rare species occupancy scales differently than common species occupancy. So unless we can come up with a reasonable way to understand how those missing rare species behave, we can't really come up with the proper shape for the species area relationship without actually looking at the rare species which make up half the species. So there are lots of different ways out there in the literature for trying to upscale from point samples to the whole species area relationship. But the real question is, do any of them actually work? It's easy to come up with lots of different ideas for doing something. Cyrano de Bergerac, in Rostand's play, came up with 7 different ways he could have gone up to the moon. He could have sat on a metal plate and thrown a magnet in the air. He could wet his hair with seawater and be pulled up with the rising tides. Having lots of different ideas of how to do something doesn't mean that you can actually do it. What we need to do to test these models is to actually find a good data set. To upscale, we need at the very least a representative set of samples, which are themselves an unbiased subset of the greater area we're trying to understand. They could be randomized or systematic or a stratified random sample. And then we also need some sort of comprehensive survey method so that in those samples we know all the species that are actually there. The best sample I know of is something called the Countryside Survey, which is done in the UK. It uses a sample of 569 1-kilometer cells scattered around the UK in a stratified random manner. In each one of those square kilometers, it sets down 5 X-plots, they're called, each of which is a multi-scaled sample itself. It has subplots that are 4 square meters, 25 square meters, 50 square meters, 100 square meters, 200 square meters. And each one of those, they measure how many different species of plants are found. And it's done by good botanists, they get them all. Now those X-plots aren't quite in random spots. They set them up in places where they don't cross habitat boundaries, which arguably makes them a little bit less biodiverse than a truly random sample would have been. So we can also take those plots and add to them the nearest linear plots, little 1 by 10 meter plots that are in linear habitats, such as hedgerows or stream banks. And add those together into an X plus linear sample, which arguably should be overestimating biodiversity. So we have 2 different sets of samples, one arguably underestimating it, and the other one arguably overestimating it a bit. To test the robustness of the methods, we need to have more than one data set. So we took the whole data set and divided it into 5 different regional subsamples, and we also came up with 10 different statistical subsamples with different subsets of the X-plots in different subsets of the sites as a way of being able to test just how general and generalizable the different methods were. And then we can compare the upscaled estimates to the actual species area relationship, because it happens around the same time as the Countryside Survey. The new atlas for the British and Irish flora was being published, and those surveys should be telling us with fair confidence the actual number of species in each of these 1-kilometer cells. This is a non-trivial challenge. Ultimately, these samples are from areas of 4 square meters up to 200 square meters, or even 210 if we add the linear plots. But we're trying to estimate the species in all of Britain. That is overall a 14 billion-fold upscaling. Even if we look at all the plots combined, we still have a 500,000-fold upscaling from the whole sample up to Britain as a whole. So this is a non-trivial challenge. So for instance, if we just simply took the finer scale data and extrapolated it linearly, you can see we'd missed the real species area relationship by quite a bit. So how did the methods do? Some of them have produced extremely high estimates, greatly overestimating the number of plant species in Britain. Others produced very low estimates. Some of them predicted that there will be fewer species in Britain than there actually were in the data set they were using to fit the models, which when you think about it is a very bold prediction. Other ones got the total number of species more or less right, but had the shape of the curve quite wrong. Other ones got the curve shape pretty close, but didn't have the right number of species. Overall, we can try to put together all the results of all the different models into a great big table. And in fact, 2 different tables, one looking at species richness and the other looking at curve shape. So this is the table for the species richness, and one model stands out. The Shen and Hay model ends up doing a pretty good job of predicting the actual number of species in Britain, and it does almost equally well whether it's using the 4 square meter plots or the 200 square meter plots. It doesn't seem to take much data to get a pretty good estimate of how many species there are in Britain from these samples. The Chow-2 method, on the other hand, while it's well correlated with the right answer, tends to systematically underestimate how many species there are in Britain by a factor of 2. But that's still pretty good compared to some of the other methods, which either greatly overestimate biodiversity or greatly underestimate biodiversity, or in the case of the canonical power law, produce estimates which are all over the map, but completely uncorrelated with the actual answers. Now, as the shape of the species area relationship, that's a much harder challenge. 2 of the methods end up looking pretty well. One of the methods from Kang Hui at the University of Stellenbosch ends up making a pretty good prediction across the wide range of scales of the shape of the species area relationship and its height for the UK. An even better estimate comes from a method suggested by Arno Schissling. Arno's method works not by upscaling from a fine sample, but actually downscaling from an estimate of the total species richness. So here we have Arno's method Downscaling from the Shannon-Hay method, we already showed did a good job of predicting total species richness. As you can see, once you've got that estimate of total species richness, you can downscale very effectively to understand the rest of the curve. Other methods don't work quite so well. So for instance, the power law method, which would have been the way we would all have attacked this problem back in the 1970s, really is all over the map. Which brings us back to Cyrano. We have a lot of different methods, and the question is, do any of them actually work? One of his 7 different ways of getting to the moon was a rocket formed like a giant locust with saltpeter explosions from its rear. Is that really so different from a rocket ship? In fact, Arthur C. Clarke argues that Cyrano, not the fictitious Cyrano of Rostand's play, but the real Cyrano in the 17th century, basically invented the rocket ship as a concept. So sometimes a crazy idea actually does the job. In summary, biodiversity is inherently scale dependent. Not only that, but biodiversity change is scale dependent. And it may well be that some of the drivers of biodiversity change that we're imposing on the planet will affect biodiversity differently at fine scales, at moderate scales, and at global scales. In principle, it ought to be possible to estimate biodiversity across the full range of scales using locally monitored biodiversity data. We tested a range of those approaches using a high-quality data set from the UK. And we found those models differed a lot in how well they performed. The best approaches, though, performed pretty well. There's still plenty of room for improvement, so novel approaches to upscaling biodiversity are still welcome. But I think it's fair to say that at this point, biodiversity upscaling has graduated from being an intellectual plaything to being a practical tool for biodiversity monitoring, modeling, and ultimately conservation.